Hi, welcome to Neuroverse
A podcast hosted by Carolina.
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on different animal models in science. I chose this topic because recently I've been speaking to a lot of my non-science friends about what I do. And whenever I bring up the fact that my main current model organism that I use to do research is a mouse, everyone always gets super surprised. How is mice relevant research to humans? Is it even translational? And so then I have to explain the whole spiel of fundamental science and research, and then clinical science and research, and then potential pharmaceutical applications and the whole process. And so this inspired me to do an episode and focus on the wide variety of animal models that we have in science and how each bring something incredibly useful and unique to the table. And I also want to point out that it's not just necessarily the goal to be translational to humans. A lot of it is just to have a very fundamental understanding of the world around us and how mechanisms work and laws that apply to all biological systems.
Yeah, I can definitely relate to that as I also mainly work with rodents. But yes, preparing for this episode, I think I realized how neuroscience's foundation is in biology. And what is our goal in understanding neuroscience? Is it consciousness? Or is it also just understanding how different animals work? And there's a lot of behaviors that are specific to subsets of animals that aren't necessarily translatable to humans, but it's still informative to understand the neural circuits that contribute to them.
Yeah, definitely. So there are several animal models used in science. The biggest ones is rodents. They make up approximately 95% of all laboratory animals. And then there's also yeasts, which are useful for biological studies of cell processes such as mitosis and even diseases such as cancer. Then there's also C. elegans and roundworms. They have a very fast reproductive cycle, which is very beneficial when working with animal models. The neurotransmitters that control a lot of their behaviors are conserved from worm to human in about 40% of diseases. And they are virtually transparent, so they can be visualized with a microscope and some fluorescent proteins. And then this is a similar story for Drosophila fruit flies in that it was the first fully sequenced genome, and about 75% of disease-related genes in humans also have functional pathologies in flies.
Also, one of my favorite neuroscience fun facts is that the circadian genes, also known as the clock genes, which got the Nobel Prize, were actually discovered in Drosophila.
Yes. And then there's also zebrafish, which are transparent, which have incredible microscopic utilities. And there's some really cool technologies that using 2-photon calcium imaging, we can visualize the neurons. So using a really powerful microscope, we can see the neurons having dyed the neurons, and we can see in real time zebrafish's behavior and the signaling. And so I think these are some of the biggest ones. And there are other smaller, more niche, I guess, unique animal models. So one that caught my eye was Professor Lars Tschitke at Queen Mary University did a study looking at dancing bees and how bees communicate. So they gave the Bombus terrestris, which is the bumblebee, a puzzle box task, and the bees were challenged to opening this box in a way that was a non-natural foraging behavior. And they tested to see whether if they introduced a bee that had been previously trained in this task, whether the newbies could learn faster or if they learned slower. And what they found out was that the bees that observed another trained bee opening the box and solving the solution, that they were then able to learn faster. And this suggests that social learning is crucial to understand the box opening. And they also showed that some bees were able to spontaneously open the box, but they were much less proficient than the other bees. So this is really interesting because, I can already hear my friends' voices in my heads like, why are you doing social studies on bees? They have nothing to do with us. How are they even social? And this just goes to show how social learning played such a crucial role in their survival, clearly. And so we can observe these behaviors and observe the biological mechanisms underlying these behaviors and how they translate to us.
That's so cool. And yeah, I think bees and other insects are really good model organisms specifically for social behaviors, which I guess, yeah, sometimes we have a tendency to think is a human specific thing, but I mean, they have colonies, right? And I also came across a bee paper called Psychophysical Laws and the Superorganism. So referring to the whole colony as the superorganism. And it was so interesting because it basically compared the workings of a bee colony with a single brain in a higher order species like a human. And so they looked at psychophysics, which is the relationship between the intensity of a stimulus and its perception. And they used a series of psychophysical laws to augment a mathematical model of a bee behavior. So specifically, they looked in these European honeybees, which they reproduce in the springtime, and then the queen leaves the nest with all of its thousands of scout bees, and then part of the swarm protects the queen, but another part goes out to scout to find a nesting location. And the scout bees, they basically return with a potential nest site. And then the other bees need to agree that it's worth going to that nest site. And they can send a stop signal if they don't agree. And then you need enough bees agreeing. And then they do this waggle dance. It's very interesting reading about it. And basically, they found that so they used the mathematical model of nest site selection that has already been defined for this type of bee. And then they changed the parameters, specifically the amount of information that needs to be processed and the stimulus intensity. So the nest quality. So they showed basically that the colony decision time increased with the increase in information that needed to be processed, which was the number of available nests that there were. And the colony decision time also decreased when the nest quality was worse. And they basically showed that a combination of these two established laws, known as the Hick-Hyman law and the Piron law, could determine the decision time of the colony. And then they concluded that altogether, these theoretical results that could predict the behavior of the superorganism or the bee colony is comparable to that of a single brain, and that there are fundamental laws that arise from these mechanisms of information processing and decision making. So I think this is a really good example of how, although it's quite a rough description, I think, of the behavior, and the comparison isn't direct, it's more inferred. But I think it's a good example of how understanding behavior of different organisms can actually be applied to understanding also the human brain.
Yeah, that's so cool. The way you were describing it definitely reminded me of like an organism, and that like the multicellular parts make up the whole organism. And it also reminded me of our episode on the individuality and what makes up an individual. Yeah, super cool. And I like the I guess cost benefit analysis that they make and how it really relates to decision making. It's really interesting. Also, I just remembered relating to what I was saying regarding bees and being able to solve a puzzle. That reminded me of our episode on the blob, and how that's another example of another very simple organism that was able to solve puzzles, and that studying the mechanisms and I guess studying its evolutionary behavior teaches us a lot about, I guess, the most efficient ways of mapping a tube system or solving a puzzle, and how things like habituation can be present in many different types of organisms. And we don't need to always revert to larger organisms. And this relates to the 3 Rs, which is replace, reduce, and refine. So the 3 Rs are something that is quite well known in the animal testing and ethics community. It's a principle installed in most governments and scientific bodies that use animals to carry out their research. And it's a way that we can ensure animal rights and well-being are taken into consideration. And so these agencies promote this principle called the 3 Rs, which is reduce, refine, and replace. So it's always good to move towards that. I have one more paper on bees, and then I promise we'll move on. So this next one is just about how bees learn to dance. So bees have a form of communication that allows them to send their nest mates to the location of food source by using symbols, which is similar to what you were just
Yeah,
talking
that's
about.
dance.
Yeah, the waggle dance, exactly. So yeah, so the way that they do this is they encode the coordinates in the movements, and they do this dance on the vertical wax comb in the hive. At least that's what I read in this paper. And the motions of the movements are picked up by recruits who observe other bees dancing. And only these are then able to pick up the movements and learn to communicate this way. So this communication system is once again learned socially. So the waggle dance consists of performing a figure of eight, and the duration of the waggle dance and the waggle run informs the others the distance to the bounty, so to the food. And then the direction of the target is relative to the sun encoded in the angle of the waggle. So they encode,
Whoa.
yeah, so they encode the distance and the angle. So a waggle run straight up means fly towards the sun's azimuth. And the waggle run at an angle at 20 degrees to the right means fly 20 degrees to the right of the sun's azimuth. So, yeah.
That's pretty complex sounding. That sounds like a thing humans did back in the day.
Yeah. Yeah, true. And it's interesting you say that because in the paper, they often compare this as a type of pre-programmed language in the same way that humans learn new words through symbols and meanings. They compare the ways the bees learn these symbols and meanings. However, I have a counter to this, which is they say that the waggle dance is innate and that there's evidence that it's genetically encoded. But then if that were true, then it wouldn't be a completely socially acquired thing. So if they never witnessed the behavior, they wouldn't really be able to reproduce this, which I guess is also similar to human language.
Yeah, there are innate forms of it or innate aspects as well as learned aspects. I was actually thinking about that as you were speaking. I was like, I wonder if they just naturally know exactly how to move or if it's learned. And then you said the thing about social learning. So I guess it is a combination.
Yeah.
I actually have something to say that's really related.
Tell me, yes.
So another model animal that's really useful for understanding vocal learning behavior is songbirds. Yeah, there's also innate and learned versions of vocal behavior. So I was reading this review article about all the similarities between songbird and human systems for vocal learning. The interesting thing is that they're evolutionarily quite far apart. So they actually evolutionarily develop differently. So, for example, primates who are evolutionarily very similar to humans don't have a vocal learning behavior, but songbirds do. But yeah, basically the paper explained that humans, as a human would know, can imitate other sounds. And we use this auditory feedback system a lot to learn how to speak and to communicate. And this process is very similar in songbirds, where there is also this participation of auditory feedback with young songbirds as they're learning songs. And another important feature of human language is to have flexible control over syntactic structures, so reordering words in a sentence. And similarly, it's been shown that certain finches have the ability to control the ordering of syllables in a song. And then in terms of actual biology, there are also analogous cortex to basal ganglia to thalamus to cortex circuitry that's associated with vocal learning behavior, suggesting that there's also a similar neural basis.
That's super cool. I'm so glad you brought songbirds up. It's so fascinating how evolutionarily separated so early on, but then our languages, they're so homologous as well.
Yeah, I guess it just shows the nature of evolution because it depends on the evolutionary pressures. And also, it shows that evolution is really just finding the best solution out of all of the random mutations that happen and the random outcomes you get. And then you just select for the one that works best. So it may, like humans may not have developed a language, but we did. And then I guess the fact that the neural circuitry is analogous, it shows that's kind of the optimal neural circuitry as well for this vocal learning. Yeah, so cool.
Yeah.
I came across another study that is related again to insect behavior, but not bees. This time it is ants.
Nice.
So, the study by Sheng et al. From the University of Pennsylvania that came out in 2020 is called Social Reprogramming in Ants Induces Longevity Associated Glia Remodeling. And I think this study really showcased well a specific technology, which is RNA sequencing, where they looked at the RNA in different social castes of a species of ant called Harbagnethos saltator. And so the unique thing about ants is that they have these social castes where they carry out distinct specialized tasks like reproduction or foraging or defense. And then these different castes also have different life expectancies. And specifically, the reproductive queens tend to live longer. So the unique thing about this Harbagnethos saltator ants is that instead of developing their caste in the larval stage, they actually develop it later on in adults. And it has been shown that these different castes have different neurobiology, but it's unknown whether these differences occur at the cellular molecular level. So that's what they looked at, and that's why they carried out single cell RNA sequencing from the brains of these worker ants as well as the queens, which are called the gamer gates. So then what they found is super cool. They showed that during the transition into the Gamergate status, they had a 40% expansion in ensheathing glia. So there were differences in the glia cells, not the neurons. And then the cool thing is that ensheathing glia are essential for brain health because they remove structures generated during stress or injury. And then they confirmed that these ensheathing glia do upregulate transcriptional programs in response to brain injury. Then, on top of that, related to the life expectancy that is increased in the gamer gates in these reproductive queens, they showed that in the worker ants, there was a decrease in ensheathing glia during their lifespan. So it's kind of like a type of neurodegeneration, I suppose. But in the gamer gates, there were no changes in these ensheathing glia. So they likely contribute to the increased life expectancy. And they mention in the discussion part of the paper that this could be, a really valuable target for future research on anti-aging and increasing the life expectancy in humans that could potentially be translated, even though who would have expected that you would have found this from looking at a mechanism to differentiate social castes in ants? I think it's super cool.
That's amazing. I love that. Yeah, this is exactly like the crux of why I love fundamental science, and that you just go out and you figure out the mechanism and you probe, and then lo and behold, a lot of it is translational and has such incredible uses.
Yeah, you can always find some use to the knowledge that you gain.
Mm-hmm. Mm-hmm. So there's another model organism I wanted to briefly touch upon, which is the social amoeba dictyostelium. So this is a unicellular organism, which you may not have expected to be used as an animal model. How can it be, useful for human related research when we are multicellular and we have a lot of complex behaviors, etc. But one of the strengths with using social amoeba is that it has a lot of conserved proteins that we also contain. So regarding studies looking at single cell processes, and because a lot of the protein that are conserved in the amoeba and this unicellular organism translates across the whole evolutionary chain, which shows that these proteins are important. So if something goes wrong in these proteins, okay, this is a good place to start looking at why it went wrong. So one key thing that they found in the social amoeba, this research was carried out by Professor Robin Williams, is identifying how valproic acid works in the amoeba and in the brain, which led to potentially novel treatments for epilepsy, as well as looking into a drug treatment used in epilepsy called the MCT ketogenic diet. So the name may sound familiar because of the ketogenic diet, but this is looking specifically at the fatty acids that this diet focuses on. So one of the useful things of using this model is they're able to screen lines and screen overexpressed proteins, or, conduct a lot of biochemical analysis that are not usually available with mammalian models, and also other tools that are not available for mammalian models. So they looked at the mechanism of action of sodium valproate, which is the most commonly prescribed treatment for epilepsy, and they identified a medium chain fatty acid that is a key component to regulating seizure control. And so they were able to research this using the social amoeba, which can then be translated to humans. And so all of these animal models and many more have so many powerful utilities, not only for humans, but also just for understanding biology. And there's this argument that always comes about in terms of anthropocentrism regarding the study of animal culture, which is inevitable. And some look for what makes us different to non-humans, whereas to others, the appeal is the chance of finding ourselves staring back. So whichever end of the spectrum you're on, animal models are great to study.
Yeah, and I think the most important point, as you mentioned, is the fact that the tools that we have guided what animals are being used, and then it's kind of circular. So then because this animal is already used, then we make tools for it. But mostly, like, to justify the ethics of using mice, for example, it's because there's a whole host of genetic tools that we now have, all the different mouse lines that we can genetically encode, fluorescent indicators or calcium indicators and beyond. And then also look at the behavior. So it's always like a balance of the advantages and disadvantages. And as we've covered throughout this episode, each model organism has its own perks.
Yeah. And I think that's a great way to end today's episode. Do let us know if you work with animal models. If you like this episode, please feel free to share it on social media, Twitter, Instagram, Neuroverse_pod.
And check out our website for more information. And feel free to fill out our feedback form, which is linked below. And I hope you enjoyed the episode. Thank you for listening.